We just talked about Mendel. We talked about how he looked at these different plants, each of which had a character that had two distinct traits, and he mathematically studied how those 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 traits were able to be predicted in future generations. Now, he didn't know anything about modern molecular biology. He had no idea about how the cell may have actively reproduced like we do, knowing that there are two units of inheritance in the homologous pairs. So in this lecture, what we're going to cover is Mendel's two laws of inheritance, and we're going to connect that to what we learned in previous lectures about meiosis.
There must be two distinct units of inheritance. One that would contain one trait, one character of a trait, and one that contains the other. Because when you pass them down, one can mask the other, dominant will mask the recessive, but as they get passed down the second time, you end up with an independent assortment event. Now, Mendel knew absolutely nothing about modern genetics. He didn't know about chromosomes or anything like that. But he understood that there must be a unit of inheritance. What we know of as the unit of inheritance is the chromosome. And how many chromosomes do we have for each unique chromosome? We have a total of 23 unique chromosomes, but we have 46 because we're termed diploid, right? We have two distinct chromosomes that encode for roughly the same thing. We have what are termed homologous pairs. So that distinction between one gene and another became known as alleles. An allele is a variant form of a gene. That's it. Slight difference. Instead of being GATCATTA, it might be GATTATTACA.
So why do we need them?
Why do we need them? They lead to differences. So slight differences create, let me show you real quick. So we have a slight difference in the DNA. There's that C was replaced with a T, which means that now instead of being CUAGUA, it's CUAAUAA.
So that's the allele. Allele of that gene?
These two are different alleles of the same gene. So now the amino acid sequence is no longer tyrosine and lysine. It's now tyrosine and, alanine. And because of that, back to primary, secondary, tertiary, quaternary structure, it's going to fold differently. You're not going to have the same chemical connections, which means that you're going to have a different shape, a different form. And if you have a different form, what else do you have? Different function. So let's say this creates a purple allele, a purple trait, a purple pigment. That's what we'll just do, purple pigment, and I'll spit a lot. So we have a purple pigment here. This one has a totally different shape. This one might not function at all because of that one change, which means instead of being purple, this one's white. So two different alleles for the same trait. They're located at the same place, at the same locus on the chromosomes. Imagine this. Each cell in this flower has two chromosomes in it. One is purple and one is white. What color will that flower be? Purple, because you've got that purple pigment there. It's producing purple pigment. 3 P's in a row. So you've produced purple pigment. What if you have two copies of this allele? This particular one. You've got purple and purple. So what color is the flower? Purple. Here you've got purple and white. So what color is the flower? Purple. But what if you have two of these? Right, because there's no purple. Correct. Yes?
So you're saying the recessive trait gene doesn't show up until the F2 stage?
Correct. If you cross two pure breeding plants together, it won't show up to the F2. Alleles are really important for studying biology. Alleles are actually hyper important because without alleles, you'd never see differences. These differences will lead to different reproductive rates. A purple flower might be better at reproducing than a white flower. And if that happens, then the frequency of that allele in the population goes up, and that's where you start getting into population genetics. That's where you get into gene pools. Now, I'm big on alleles. Did I tell you guys the story of my daughter's birth yet? Let me tell you the story of my daughter being born. All right. So one of the most important decisions you can make in your life is what you're going to name your kid, because you can mess them up for the rest of their life. Now, I mean, yeah, oh yeah, you totally can. I cannot tell, and Riley's not here right now. In Chesterfield, for some reason, in your generation, Riley was a huge name. I don't know why. There's like 50 of them in my classes. They're all over the place. I didn't want that for my kid. So my wife, who we've already agreed to is the dominant one in the relationship, decided she wanted that we were going to have a girl, her name was going to be Kira. And I said, oh, that's cool. You like that. I'm good with that. But I want her name to be Allele. I know, right? And so I was like, that sounds great. And she's like, no, that's a bad idea. Her name will be, I think it was like Kira, was it named Catherine after her mom? I don't remember. And I said, no, I really like Allele. And I was studying graduate school. I thought it would be really cool because then she could grow up and be a biologist and be like, Allele's my middle name. Literally, yeah. So my wife vetoed that hard. She was like, absolutely not. And she won. So we went to the hospital, and she wanted to have a natural birth because it was her first one, and she didn't understand what pain meant. And then she felt pain. And I didn't have any insurance at the time. We were paying totally out of pocket. That was a $25,000 kid. And the one bill that I did not mind paying was that anesthesiologist because he knocked her out. Yeah, epidural, and she was all happy after that. It was great. She had the girl came out, and Kirsten went to sleep. And then they came in with paperwork. And they said, are you the father? And I was like, yes, I am. All right. Can you fill out this birth certificate? And I was like, yes, I can. So now her name is Kira Allele, which is great. So it's Kira and Allele, and that's me, Pantasis. She was just happy it was over, I think. And she's gotten over it since then. All the time I call her Kira Allele, especially when she's in trouble, which is really, she's a teenage girl, so it's like all the time. But no, usually it's just Kira. But, yep. What's great is she learned cursive, and her name is... I know. I read your exam. So that's the story of my daughter getting this name. Oh, and then, of course, if we ever are out and you see us, and she notices that you're one of my students, she will absolutely judge you. If you see a little dark-haired girl, her hair's down to her waist, and she's staring at you, it's because she is judging you and thinking, do I know more about biology than they do? Yeah. Nah, well, she used to.
Wait, how old is she?
13 now. So, yeah, that's always fun. She judges her teachers that way, too. They call her on the roll, and she's like, my name is Kira Allele Pantasis, and if they have no reaction, she's like, they're not worth it. So there we go, alleles. Alternate forms of genes. The alternate forms of genes that allow us to study genetics. They allow us to have a competitive edge. They allow us to ultimately learn more about the world. During gamete formation, so meiosis, you're going to have the segregation of alleles. That is, you've got your tetrads, and the homologous chromosomes are going to segregate independently. That means if the mother's chromosome moves to one cell, that doesn't impact whether another mother's chromosome goes with it. You could have, back to our fingers, All right. So you could either go this way or you could go this way. It is totally random. 50-50 shot. Right, we were talking about Mendel. So we've got we've got the law of segregation. This is the law of independent assortment. Here we have alleles of different genes assort independently of each other during gamete formation, if they're not linked, that is, they're not on the same chromosome. So we've got 2 chromosomes here, 2 tetrads. This tetrad, that tetrad. You could have this A and that big B go together, and if that happens, the little a and the little b go to either end of the cell. Does that make sense? You could have it go just the opposite. A big A and a little b, and a little a and a big B. Equal chances for both. You know what? I'll draw it out and show you even better. So it is possible, absolutely possible, to have 2 green ones go together and 2 red ones go together. Does that make sense? Totally possible. It is equally possible to get a red one and a green one, and a green one and a red one. So what are my chances of getting any of these? 25%, or 1 out of 4. Or 1 to 4.
Doesn't matter how we write it.
Nope. I can interpret a number as a number as a number. When we're studying these, when we're studying alleles, what we're ultimately looking at is how the traits are inherited. What we can see is the phenotype. The phenotype is the physical expression of the trait. It's the result of genetic expression. Tall, yellow, green, all of those characters that you see are the phenotype. This is a phenotype. This phenotype is syndactyly. Syndactyly is fused digits. Polydactyly is a phenotype, an extra digit.
What is it when, like, 2 people are in one body, you know, with the 2 different heads?
Conjoined twins?
Yeah, is that like the same kind of thing?
So conjoined twins happens due to developmental abnormalities more than genetic expression problems. But we can go into that later if you want. And you're just like, follow.
Yeah. They play volleyball together, but they each control one side of the body. It just blows my mind. Yeah, it's so
So we've got the phenotype is what you see. The genotype is what's actually in the genes, what's actually on your DNA. This DNA, that's a genotype. This is a genotype. The genotype are the molecular instructions. We do not want to write out this big long sequence, especially because these big long sequences are usually about 1,000 base pairs or longer. So instead of writing the whole thing, we just give it an arbitrary capital letter to represent this. So we might say that this equals, I don't know, sure. And then this one down here equals, actually we'll go with the little p, because this is dominant to that. The capital, giving it a capital letter usually represents dominant. Recessive alleles are usually represented by a lowercase letter. It is totally up to you what kind of letters and things you want to use. All that matters is that you properly define them. Heck, you could use colors, you could use smells, you could do whatever you want to define these things. Now I've known people, they use different types of markers that have different scents. No, seriously, geneticists get weird. I didn't do it that way. But other people, it worked. Typically, the best letters to use, actually, here are the letters to avoid using. C, I, J, K, O, I know, I didn't mean to. U, E, W, X, and Z. Those are typically the ones I would avoid, but you know what? I'm not you, and it doesn't matter in the end. You have to be able to tell the difference. When I do capital and lowercase and I have to use something like that, I'll typically put a line under it just to indicate. But again, you can use different colors, you can use different letters, you can use whatever you want. There is no standardized way of doing this. Except maybe on your labs. I don't know if you have to do a multiple choice thing where whatever. But when you work out things using these, because you know you're going to have essay-ish questions like this, short answer questions where you're going to have to solve a problem I give you. If you were to identify your alleles first, I'll at least know what you're trying to do. So here we've got a moth. This capital A represents a dark allele. So if you have two dark alleles, what color is it? Dark. You have one dark allele and one totally broken allele that doesn't work. Dark. Two totally broken alleles? White, because you were not producing any pigment. Well, now we know we have two distinct units of inheritance, a dominant one and a recessive one. When you have the exact same alleles on both of the homologous pair of chromosomes, so the exact same allele, that's termed homozygous, because homo means same. Same zygosity. When they have different alleles, two distinct alleles that are going to encode for different versions of the same gene, that's termed heterozygous. Hetero means different.
There's a lot going on in these Mendel lectures. In this particular group of lectures, we talked about alleles as variant forms of genes. So you have a gene, and then its variants. If you've got two variants of the same gene, that would be an allele. Every diploid organism out there has two homologous chromosomes, each of which has the same gene at the same locus. They have the opportunity to have two different alleles. So if there are differences between genes at a locus, those genes are known as alleles of that gene. Talk briefly about the law of segregation, which states that every organism can have two alleles for each trait, and that those alleles separate during meiosis, giving each gamete a single allele. When those gametes fuse back together, that means the offspring will then have one allele for each trait of the parents. Alleles of different genes can assort independently of each other as well, with the law of independent assortment. So that no two chromosomes are linked together. The genes on two chromosomes are distinct, and they're going to segregate independently of each other. That is, without regard to the other chromosomes. We're going to look at genotypes and phenotypes. Genotypes are what alleles are present in an organism, and phenotypes are what trait is actually expressed. So alleles are what are the genes. Genotype is what are the genes, and phenotype is what do you see. That genotype could be heterozygous. They might have different alleles. Or it could be homozygous with the same alleles. And we'll talk about applications of that in the next mini lecture. These content review questions are here to really focus your studies, but going forward into the next set of lectures, we're going to take what we learned in the first mini lecture and the second mini lecture about Mendel, and we'll start applying it to inheritance patterns. We're going to look at Mendelian inheritance. We're going to work out some problems and see if we can predict, based on what we know about parents, what the offspring will look like.